Thank you very much, Tony, and thank you for inviting me today to have a chat to you all. I'm going to try to share my screen. It worked before, so hopefully it will work again just fine. And okay, so I assume that we can see everything now. All right, so I'll get started then, unless someone yells out that they can't see it. Okay, so I'm going to talk to you today about the assembly and dysbiosis of seaweed microbiomes. And I've decided to break this down into two parts. So the first part of the talk will be discussing microbial communities that assemble on seaweeds and how they assemble on seaweeds. And the second half will talk about bacterial diseases in seaweeds and what we're starting to understand about how we can use microbiomes for disease mitigation. But unfortunately, I believe that many of you might have to be convinced first as to why we might want to study seaweeds, and that they're not just the smelly things that wash up on the beach or get tangled in your fishing line. But in actual fact, they are very diverse organisms that are really important as ecosystem engineers across many coastal habitats, where they provide food and shelter and breed grounds for many other organisms. Now, as an example of the ecological importance, we can consider Australia's own Great Southern Reef. This is a temperate and seaweed-dominated reef that spans many thousands of kilometres along the bottom coast of Australia. And like its northern neighbour, the more famous Great Barrier Reef, it is a spot of really high biodiversity. And if we then consider its economic value that is generated through the number of different things like tourism and fisheries, it's estimated that it's worth over 10 billion Australian dollars per year. So seaweeds don't just have an environmental value, they also have a growing value in the aquaculture sector. They're one of the most growing aquaculture species in the world globally, with interest obviously in food production, biopharmaceuticals, biotechnologies, but there's also a growing interest in growing seaweeds for nutrient and carbon sequestration. So overall, it's estimated that this economic value of seaweed cultivation alone is over 11 billion US dollars per year and growing. So these seaweeds are, of course, not living alone, and we're all here to hear about microbiomes. Seaweeds also harbour a very rich biodiversity of microbes. And while they're not just bacteria, bacteria have predominantly been the ones that have studied the most, and it's what I'll focus the talk on today. So bacterial densities across seaweeds can range from maybe a few hundred cells per centimetre squared on what's called the thallus, up to maybe 10 to the 8 bacterial cells per centimetre squared. And they, just like their abundance, their diversity can vary according to different species, different space along the actual organism itself, biogeography, as well as different environmental stresses and seasons. So these microbes are not just redundant and sitting there. We now know that they play an important role in all different life history stages of seaweeds. This was kind of known already sometime way back in the 1980s and beyond, when researchers trying to grow several species of seaweeds azenically, so without their microbes, found that they were not able to form their proper morphologies and structures until they added microbes back. We now know that there are certain microbial species that are important for early settlement, so providing settlement cues to the spores, for example. They're involved in morphogenesis stages, outgrowth into adult seaweeds. And then along the surface of the seaweeds, there's microbes that are providing supply nutrients and act also as kind of like a second skin, as a protective layer, and providing chemical defenses, for example, to protect the seaweed from grazers, pathogen attack, and anti-fouling organisms. So one of our focuses right now is to ask the question, how are these microbes assembled on the surface if they're so important, and where are they actually coming from? So in most microbiome type work, we can think of an organism getting its microbiome either from its parents, so vertically transmitted, or from the surrounding environment, so horizontally transmitted. And that's one of the questions that current PhD candidate Shakur is looking at right now. And he's asking whether or not seaweeds actually can inherit their microbiomes from their parents. Now, without going into any exact details on the complex life history of different seaweeds, he's taken a few what we call models from seaweeds around the Sydney area, and he's starting to investigate the potential for vertical transmission by looking at these early life history stages. So in some seaweed species, they have egg and sperm, in others, they have spores. And he's done microscopic imaging, either through scanning electron microscopy or fluorescence in situ hybridization, and found evidence in several seaweed species that we actually have microbes that are associated with those early life history stages. He's then gone on to look at using the community profiles, so using 16S RNA gene amplicon sequencing, and he compared the adult microbiota with the eggs or the spores. And he's then found that in some cases, there's evidence that transmission from parents is possible, but there's little selection. So in the case here of Phyllospora comosa and Delicia pulchra, you can see that basically all the microbes that are found on the eggs are also found on the adults, with maybe some differences in their relative, in their relative abundances. But overall, there's seems to be little selection. And in contrast, he's found that another brown alga, Homosara banksii, is actually does have evidence of transmission, but with some degree of selection, such that not all the microbes are the same. There seems to be just a subset of those microbiota that are associated with the eggs. And so this is ongoing work, and we're quite excited to see how this pans out. But one of the models that we've looked at, Ulva australis, seems to have no evidence of vertical transmission so far. So whenever he looks, the spores appear to be free of any microbes. And so this suggests that, at least in this case, these microbiomes are really dependent on that horizontal or environmental transmission. And that kind of makes sense if we think about what we've known in the past about microbiomes associated with seaweeds. So work done over a decade ago by Catherine Burke and others basically set out to ask the question, how do the microbial communities of the seawater, which could act, as we think now, as a seed bank for microbes associated with the alga, how do they differ from the host? And what they found using, again, 16S RNA gene analysis is that there was very little overlap. So only about 5% of the bacterial species were shared between seawater and the host surface, indicating some degree of what we call niche selection here. But then if we dig a little deeper and we look at the individuals, we find that even individual replicates of the seaweed were actually very variable with their taxonomy. And it appears that they only shared about 20% had common microbial species associated with them. And this suggests then a random selection process is occurring. And so to really try to understand this a little bit better, Catherine and others basically did one of the first metagenomes at the time of a seaweed host organism, and basically found that similar to their taxonomic profiles, metagenomes or functional profiles were very distinct between the host and the seawater. However, unlike the taxonomy profiles, their predicted functionality was highly consistent between the replicates. And so if we take this taxonomic and phylogenetic assignment together, it suggests that functions of this seaweed are provided, microbial functions, are provided by different taxa in each community. So that concept has actually been described in macroecology using this lottery hypothesis of community assembly concept. And that concept was first described to understand how reef fish aggregate around certain coral reef bombings. And it goes by the current premises. So organisms, in our case bacteria, with similar patterns of resource use or functions form these so-called guilds. And these guilds may or may not be phylogenetic related. Then if that occurs, community composition of a particular surface, in this case Ulva, will depend on the representatives of the guilds that colonize first. So if we think of that in this little schematic here, this is a little environment, let's say it's the seawater, and we have here 3 representative functional guilds. Within these guilds, it's different colors. you see that there are different species, different shades of the different colors. And below we have an algal surface that's freshly ready to colonize. What we see then is that as that surface becomes colonized, there'll be a random, I guess, assignment of the different species that fit within that taxonomic, that functional guild. Those will then proliferate and then dominate the surface. So what you end up with is an outcome where communities with a set of functional genes required for, in our case, the Ulva-associated lifestyle, are colonizing that surface regardless of the species composition. Right? So the next question, the current question we're looking at the moment, is we wanted to ask then, if these communities are formed by a lottery of who arrives first, can the arrival of specific functional guilds of bacteria influence the subsequent community composition? So in ecological terms, we're interested in asking the question, do specific bacteria exert what's called a priority effect on subsequent colonizers? So to study this in a little bit more detail, we're again sticking with this Ulva community as a model. And what we've done is chosen 2 different bacterial species that we know have been routinely cultivated from Ulva over the years. They're really good biofilm formers, and they produce a number of different secondary metabolites that have antibacterial activity or antibiotic activity, such as violacin, tangerine, and this TDA molecule. So we suspect that they're going to be quite good competitors, and given these characteristics, are likely to impact other community members. So taking these models, a former PhD student who's just graduated, Jadranka Nappy, she basically did a couple of manipulative experiments where she directly colonized the surface of small pieces of this seaweed tissue, either in the presence of one strain, P. tunicata D2, or the Pseudovibrio D323, or the control. She made sure that she could see that they colonized using microscopy, and then subsequently she added the natural seawater community to those samples, and then followed the community composition that occurred subsequent to that using amplicon sequencing across a period of 72 hours. So what she found was that both time, represented here as symbols in this NNDS plot showing the community structure, and treatment, represented in the colours, influenced the communities that formed on the seaweed. So if we visualise that in terms of priority effects, we can assess the changes in Bray-Curtis dissimilarity values between the treatments and the controls, that's no inoculation, for each time point. And the patterns are shown here. You can see that P. tunicata had quite a large impact on the communities that formed, so it was very different from the control communities. However, with time, this difference decreased. Interestingly, though, even at 72 hours, when we found that there was the amount of P. tunicata itself was at this time close to 0 in terms of abundance, there was still a strong impact in terms of the community was still very distinct from the control communities. And that indicates that there's likely to be a strong legacy effect of this particular microbe during its early colonization. So it has an impact even though it itself isn't really appearing to be present. These patterns were different from what we saw for the other strain, Pseudovibrio D323. There was still a significant impact or difference between the controls, so that high dissimilarity between the control sample and inoculation with this strain. However, it wasn't quite as pronounced, and we did still see that it was fairly stable across the time. So that once it changed, it sort of maintained that stability. And so if we wanted to dig just a little bit deeper, we tried to understand some of the dynamic nature of these, what we call priority effects. We performed a temporal analysis based on the fold changes in the abundance of these amplicon sequencing variants, or ASVs, that were either significantly enriched or depleted in individual treatments compared to the control. So that is, we actually wanted to assess if the changes in these ASVs, due to the priority effect of either strain, would persist will be stable or would fluctuate according to the presence of that inoculated strain. And so you can see from these pie charts, the take-home message here is that the priority effect of Pseudovibrio D323 here resulted in a much higher number of changes in the community that were consistent, so stable over the course of the experiment, shown here in the purple and the blue wedges, and that the majority of these were actually unique to the Pseudovibrio D323. So these taxa were not shared with changes that were found in the P. tunicata priority effect. Rather, the priority effect of P. tunicata seemed to actually involve a lot more variable changes. So these shown in the red and the orange segments. So if we're just solely thinking of numbers alone, these results suggest that, again, priority effects of these early colonizers can be strain specific. And so, again, if just then focus down briefly on some of those changes that were consistent, we find some other interesting patterns. So these heat maps are just showing the top 30 or so ASVs in each sample. And on the right, you have differences between P. tunicata D2 and the control, and on the left, D323. One thing to note here is that these consistent changes were mostly involving enrichment of taxa. So in other words, it looks like that there was a priority effect that was resulting in those early colonizers helping to facilitate the arrival of those secondary colonizers. And this was somewhat surprising to us because, again, remember, we chose these isolates because they're in part because they were of their superior competitiveness. We also, however, observed that it looks like the enriched taxa were phylogenetically unrelated to that initial colonizer. So in other words, just very briefly, P. tunicata, which is a gamma proteobacteria, appeared to have enrichment of taxa that were alpha proteobacteria, and vice versa for Pseudovibrio D323. So in community ecology speak, it predicts that different species can coexist where there is a minimal overlap in what's called the niche space or minimal competition between the strains. And that feature is thought to be, to some extent, related to phylogenetic distance. And so while these concepts are still kind of under debate in microbial ecology, we might be tempted to think that these patterns of priority effects here could be a result of newly arriving bacteria occupying a separate niche space from either P. tunicata D2 or D323. So that could explain why we see an overall enrichment. So next, we wanted to investigate if these observed priority effects could result in changes to functional gene profiles. And for this, Jadranka Napishi did metagenomes of that final last time point we had, and she took triplicates of each of the samples, sequenced the metagenomes, and then assessed them for different annotation pipelines, so KEGG, COG, and CASIME. We look at the result here. So we can see that the average Bray-Curtis dissimilarities between the treatments and the controls, so similar to what we saw when we're looking at the taxonomic profiles, we can see that it looks like P. tunicata always was was more distantly related to the controls in terms of its functional gene profiles than D323, and that was across all of the annotations. However, these results were not statistically significant. But what we did find is that if we then drilled down and looked at individual genes, we found that there were more gene functions that were significantly differentially abundant between the treatment with D2, P. tunicata, and the control versus treatment with D323 and the control. And similar to the taxonomic changes, we found that enrichment and depletion specific gene functions were found to be both common and unique to individual colonizers. So I'm not going to go into details, but just as a quick snapshot, for example, we found something like the type 3 secretion pathway, or genes related to type secretion pathway, appeared to be depleted in the microbiomes that formed in the presence of either one of these strains. So this is kind of interesting because we know from other studies that the type 3 secretion system pathway plays an important role in many pathogens, and it could suggest that the communities that form in the presence of either of these strains have consist of less pathogenic type microbes. If we look at those features that were uniquely enriched or depleted in the presence of Pseudomonas tunicata D2, we found, for example, there was a decrease in genes related to flavonoid-like secondary metabolism. Now, flavonoid molecules can play a role in UV and oxidative stress resistance. And it's kind of interesting to start speculating about where the habitat of this seaweed, Ulva australis, is located. And it's in the intertidal zone, where the seaweed is actually, and its microbiome, are going to be under a lot of UV and oxidative stress. And so it's sort of tempting to think that maybe these microbiomes are going to be less adapted to that host environment. And then if we look at those gene functions that were uniquely different in the presence of Pseudovibrio, we see, for example, something like riboflavin biosynthesis, which seems to be enriched in these microbiomes. And that's kind of interesting because there's been lots of studies on microalgae that have shown the importance of riboflavin or vitamin B12 to host resilience in the alga. And that resilience is sometimes, that riboflavin is thought to be provided by bacterial symbionts in microalgae. And so we're not sure if that similar things are happening yet in macroalgae, but it is, again, interesting to sort of consider that maybe these microbiomes that are formed maybe overall have more benefit to the host. So just to wrap up that first section, so what we're at now is that I like to think that diverse microbial communities are important for the host. We know that there's some evidence that these microbes can be transmitted vertically, so from parent to offspring, but that in some cases, such as our green alga Ulva species, it seems to be predominantly coming from the environment. And that those environmental microbes can be assembled determining by a lottery-based method, which provides key functional traits to the host microbiome, irrespective of the taxonomy of those organisms. And that microbe-microbe interactions that occur can result in priority effects that can have an impact on subsequent community composition and functions. So like many other organisms, we now think of seaweeds as a holobiont type organism. So not just the host organism, but the host and its microbiota as one single entity. And so this is kind of a crude segue into the final part of the talk today, where I wanted to ask the question, what happens when the holobiont is under stress? And just like us, we can think of this as a crossroad between disease and health. So when we're feeling under stress, we're likely to actually feel less healthy, and just same with seaweeds. And sadly, I think there's increasing observations of disease prevalence in marine systems and also in seaweeds that seems to be related to increasing environmental stresses, whether that's pollution, increased urbanization, or impacts of climate change. And so what I want to share with you is a story around one of my favorite seaweeds, and this is this beautiful Delicia pulchra, which is a red seaweed found off the coast of Sydney and up in Japan as well. And it actually has we've now discovered this bleaching event which occurs, and colleagues, Alex Campbell and others who were investigating the ecology of this seaweed observed over 10 years ago now that there was an increased prevalence of these disease phenotypes in the field that seemed to correlate to increased seawater temperatures. Shortcutting a lot of research into the ecology of this disease, and also we now know that this has a microbial component as well. So when the host is stressed, it reduces its natural chemical defense. That loss of chemical defense then allows for infection by opportunistic bacteria. And the first organism that we studied is this Nautella R11. And we found in a number of different manipulative experiments, lab-based experiments, where we grew small little juvenile Delicia in the lab and grew them under the presence where they had lost their defense or whether they had their defense present, then exposed them to this Nautella pathogen, we could observe damage to the tissue and localized bleaching. And Alex Campbell and others have done some lab field experiments where they used bagging experiments to inoculate individual Delicia pulchra in the field, and found that there was an increased chance of disease occurring if you had initial damage, so stress to the host. In this case, it was mimicking grazing, and you had the presence of the pathogen. So we've then gone on and we wanted to actually understand a little bit about what makes these pathogens tick. So what are the virulence traits? And like any good molecular microbial ecologist we now have the fortunate ability to sequence genomes fairly easily. And so doing a number of genomic studies and looking also at making mutational analysis, we now have found that Nautella italica has a number of different virulence traits. I'm not going to go into the details here, but I just wanted to highlight one point that is relevant to the ecology of this disease. And that is, we know that these traits appear to be regulated by bacterial quorum sensing. Some of you may be familiar with this concept of bacterial quorum sensing, but just very briefly, in our rough schematic of our R11 cell here, we have these acylated homoserine lactone signaling molecules, which are then released by the bacteria, and then by the diffusion, they come back into the cell when the cell densities are high, and they bind to this cognate transcriptional regulator. In our case, it's VAR, which then allows for the expression of different virulence genes and phenotypes. So the relevance here for our Delicia disease model is that those chemical metabolites I mentioned that are the defense molecules are actually furanone-like molecules. And these have been studied at length because they actually act as quorum sensing antagonists. So you imagine a situation where you have a healthy alga that's producing these furanones, and it's able to quieten, I guess, the virulence gene expression of these pathogens on its surface by interfering with the quorum sensing system of the bacteria. But if you have a situation where the host is under stress, it has less of these furanone molecules and then allows the expression of these virulence traits to be expressed. So this is all very interesting. And at the time, we were actually thinking we really understood the system. We knew what was going on, and we had an idea of the virulence. But at the time, we were also starting to gather a lot more information of field studies, microbiome field studies. So communities, microbial community changes that were correlating to bleaching events in the field. And what we were finding is that this Nautella R11 was not always present. And so that got us thinking, well, is there any other pathogens out there that could be actually having the same impact? And that was the work that Vipra Kumar set out to do during her PhD. And she started out by going and taking a new culture library of bacteria associated with either bleached or adjacent tissue. And she then sequenced the 16S RNA gene of those isolates and then compared those sequences to the OTUs that were found consistently enriched in a variety of other, enriched in diseased events, so diseased algae or bleached algae, across a number of different disease events. And she was fortunate that she gathered, I don't know, about 20 or so representatives of these abundant OTUs from culture-independent studies that she actually could match to her culture library. And in painstaking number of assays, and probably not giving justice to the work, she then was able to identify a number of different new pathogens, and they include Aquamarina species, Ultramonas, and Agravorans. And this was quite exciting for us because now we really could look at these pathogens with the confidence that they're likely to be ecologically relevant because they were consistently found in higher abundance in natural bleaching events. So she wanted to look at the impact of disease on the natural community post-infection in these assays. And what she found was that bacterial communities on diseased individuals were less diverse. So bleached is diseased and unbleached is healthy. They were overall less diverse, and but they were more divergent than healthy algae. So the divergence can be seen here in this NMDS plot, showing Bray-Curtis dissimilarities of samples that are either bleached, diseased, or unbleached in the triangles here, healthy. And you can see that regardless of the treatment, if they were diseased, the individuals had much more divergent type of microbiome associated with them than healthy. So these ideas are consistent with what we might consider a dysbiosis of the microbial community. And this idea of microbial dysbiosis being associated with host disease is not too unfamiliar in the human microbiome world, when we think of diseases like irritable bowel syndrome and psoriasis, all having a kind of dysbiotic component to the disease. So if we just then sort of briefly recap what we think is occurring there, under normal conditions, the seaweed and its microbiota are in perfect homeostasis, and there's no signs of stress or disease. Then if we have environmental stresses occurring, so increased seawater temperature or pollution, host becomes stressed, its natural defenses are reduced. We believe that this also then opens up the possibility of opportunistic bacteria that have their adaptive, what we call adaptive traits, increased in expression. So this might be secondary metabolite production, increase in oxidative stress resistance, and a bunch of unknown factors and functions. In Delicia pulchra, as I mentioned, this might be actually related to a de-repression of quorum-sensing type bacterial signaling occurring in that microbiome population. And overall, that leads to a dysbiotic microbial community. Once that occurs, you get increased host tissue damage, and then that opens up the space for saprophytes and secondary invasion, and then you start to see disease occurring. So I guess the take-home message for that component is that stress can change these host microbe relationships, and that disease in environmental context, and perhaps also in many human contexts, is really a result of a holobiont breakdown. And that can have severe consequences on how we might view virulence and manage disease in the future, particularly in open environmental systems and where we are expecting to see increases in environmental pressure on host organisms. Okay, so the last part of the talk, I want to actually talk about one growing area of interest in applied microbiology, and that is the development of beneficial or disease protective microbiomes. And that's sort of an area that we're trying to use Delicia pulchra, that I've just spoke to you now, as a model for seeing how we can develop these. And that's the work of current PhD student of mine, Irene Lee, and she started her project by, again, going back and culturing isolates that were prevalent in healthy individuals during disease events in the field. She then took those cultured isolates and tested for their antagonistic activity against a series of the opportunistic pathogens I mentioned earlier, and then took a subset of those and tested them and screened them for disease protective ability using a similar assay I described before. So these are like small fragments or juveniles of Delicia pulchra taken through the field. In this graph, though, what we're looking at is an increase in proportion of healthy individuals. So anything that we thought was beneficial will be sort of representative up this end of the graph. And just to briefly summarize here, what she found was that she could identify two strains that were consistently protective. One belonged to a Phaeobacter species, and one was a Pseudo-Ultramonas species. And these were, when they were inoculated along with the pathogen, could actually rescue the disease phenotype that the pathogen caused. And so what she was then interested in understanding is what are the underlying changes that were occurring in the microbial community of the selected treatments post-infection. So for this, she conducted a 16S RNA amplicon sequencing experiment and compared the microbiota between the treatment with protective strains. In this case, it was the Phaeobacter strain, BS52. And then she took related Phaeobacter strain BS23, which she thought was didn't have a statistically significant increase in protection, and she considered benign. And she compared those communities to the pathogen-only treatment. Importantly, before I show you what she found, is that she could detect the inoculated strains in the community post-experiment. And that gave us the confidence that these beneficial strains were able to become established in the community. Now, for many of you, you might think, well, that sounds trivial. You just inoculated the strains. But in actual fact, it's not that simple. And similar studies that have been done in terrestrial systems with plants and corals have actually been unable to detect the addition of those probiotic strains subsequent to inoculation. Another surprising feature that she had was that we didn't actually find any strong evidence that the presence of these beneficial strains, which we chose because they're antagonistic in lab culture experiments, were actually able to antagonize the pathogen in these in vivo experiments. So she didn't see any changes in the relative abundance of the pathogenic strain when she had co-inoculated with these beneficial strains. However, she did see that there was a difference in the Bray-Curtis dissimilarities that between, with the addition, of the BS52 prior to the pathogen, that resulted in a community that was significantly different from the community with AD1 alone, but remains similar to the control. So that is the non-pathogen treatment. So if you then narrow down and looked at what was actually contributing to those differences, she found that there were two key taxa or sequences related to two key taxa that were changing. And one was an increase in the sequences belonging to this ultramos alba, which up to the level of the control sequences, and this sequence was actually down in the pathogen treatment alone. And she saw the inverse for Cubesia marina, where this sequence is more enriched in the pathogen treatments, but was low in the controls. And with the addition of these probiotic bacteria, beneficial strains, you saw those sequences becoming down close to where the controls were. So essentially what she saw was that these protective bacteria don't seem to directly attack the pathogen, but maybe mitigate pathogen-induced changes in the seaweed-associated microbial community. So if we just think of a working hypothesis we have here, is that under normal conditions, the seaweed uses its chemical defense to protect against pathogens and maintain microbiota homeostasis. However, with environmental stress, such as higher temperatures, the alga loses its natural protection, resulting in changes to the host microbe and microbe-microbe interactions with an overall net negative impact on the host. However, in the presence of beneficial microbes, either through naturally high abundance in the individual or through artificial augmentation, dysbiosis can be prevented, resulting in a net gain to beneficial interactions and disease prevention. Okay, so the take-home message that I have for you is I hope that during this last 35 minutes or so that you actually can appreciate seaweeds are critical to temperate marine habitats and have economic value, that we can use seaweeds as models to understand microbial community development in aquatic host systems, that disease does occur in seaweeds, is complex and multifaceted, involving opportunistic pathogens and dysbiosis, may not be too dissimilar to some diseases we see in other systems, but that microbiome manipulation for disease mitigation is possible, or hopefully possible, and this can have consequences for that growing aquaculture industry I mentioned earlier, as well as hopefully protecting seaweeds in the environment. So with that, I wanted to say thank you all for listening, acknowledge the other people who have worked in different aspects of the project I've talked about now and the funding agencies. And I'll take any questions if there's time. If there's not, please do feel free to email me, or if you think of a question later, I'm happy to hear from you. So